morning, students. As we have discussed about the structure of a nucleic acid in the previous video, what do you mean by nucleoside? Nucleoside is made up of base plus sugar, and the structure of a nucleotide, that is made up of sugar, base plus phosphoric acid. In this video, we will discuss about how the two nucleotides combine together and what will be formed. When the two nucleotide, this is the structure of nucleotide, means base plus sugar plus phosphoric acid. This is the another structure of nucleotide, means two nucleotides are combined together and that will form a dinucleotide. The nucleotide of third carbon and the second nucleotide of fifth carbon having the phosphoric acid is present, that will involve the loss of water molecule. After heated, it lose the water molecule, means that third and fifth carbon atom of pentose sugar that are linked by a phosphodiester linkage. Means this is a important questions arise from here. When the two nucleotides join together, what type of bond is formed between these two nucleotides? So its bond is phosphodiester bond. Now the next topic is the structure of DNA and RNA. It forms two types of structure, primary structure and secondary structure. Primary structure is formed by the nucleotide chain. One nucleotide that combine with another and it will form a long chain. Nucleotide means base, sugar plus phosphoric acid. All these components are combined together and it will form a nucleotide. Now the next is secondary structure of DNA and RNA. It consists of polynucleotide chain. In polynucleotides, the nucleotides chain are folded and it will be arranged in a double helix manner, etc. In primary structure, the sequence of nucleotide in the chain of nucleic acid, this is called as primary structure. But nucleic acid have secondary structure also. So here we will discuss the secondary structure of DNA. In the structure of DNA, it is the double helix structure of DNA. Double helix means the two nucleic acid chain that are wound about each other, and these are held by the hydrogen bonds between the base pairs. Hydrogen bonds are present here like A, T, G, C, A, T, adenine, thymine, guanine, cytosine, adenine, thymine, guanine, cytosine. Here two strands are complementary to each other because hydrogen bonds are formed between the specific pair of bases. A combined with T and G combined with C. In between the adenine and thymine, double bond is present. In between the guanine and cytosine, triple bond is present. So A pair with T and C pair with G. Or you can say that G pair with C, T pair with A. And its name is a double-stranded alpha helix. This is the alpha form, spiral form, and double strand means two strands are there. So it is a double-stranded alpha helix structure. Now the last topic is the secondary structure of RNA. RNA is single-stranded These helix are present in which only single stranded. Sometimes they folded back. They folded back on themselves and to form a double helix structure. RNA is of three types: messenger RNA, ribosomal RNA, transfer RNA. Now the question arises that what is the main function of nucleic acids? Nucleic acid have DNA and RNA. What are the main function? First, DNA is the basis of heredity, and it is also called as reserve of genetic information. But RNA help in the protein synthesis. Next function, DNA is responsible for maintaining identify the different species of organisms. And DNA is able of self duplication during the cell division and identify the DNA strands that are transferred to the daughter cell. The message for the synthesis of particular protein that is present in DNA. RNA only help in the synthesis of protein. Now the question arises is that is the most important question. What are the important structural and functional difference between the DNA and RNA? First, structural difference. Sugar present in DNA, that is a 2-deoxy D-ribose. The sugar present in RNA, that is a D-ribose. Second point, DNA contain thymine, adenine, guanine, cytosine, but RNA contain uracil, adenine, guanine, cytosine. DNA has double stranded alpha helix structure, but RNA has single stranded alpha helix structure. DNA molecule is very large, RNA molecule are very small. Now the functional difference between DNA and RNA. Point number 1. DNA has a unique property of replication, but RNA does not replicate. DNA control the transmission of heredity effects, but RNA control the synthesis of protein. Now one more important question from this chapter. What are enzymes? Enzymes are the biocatalyst. Each chemical reactions require a different enzymes. Enzymes are very specific and efficient in their action. So they are required in a very small quantity and work at an optimum temperature, that is 310 Kelvin, and the pH that is 7.4. Thank you, students.